This is a production of Cornell University.
I'll be focusing on presenting some of the stuff that I've been doing within the Sorrells lab over the past few months, and that has been malting barley water sensitivity. So a little bit of an outline. We'll give some context. What is water sensitivity? Why is it important? And how is it measured? Because that kind of helps a person conceptualize what it actually is. Some historical literature about it, populations that have been using to answer some of the questions that we've asked, and the results and conclusions thus far, along with future steps. So, some context. What is water sensitivity? So, it's one of the traits that contributes to malting quality. It's basically a reaction to steeping conditions that causes a differential germination of malting barley under different water concentrations. So essentially it's an induced dormancy or a secondary dormancy. So after the dormancy has been broken after the original dormancy has been broken, exposure to certain steeping conditions can cause the barley to have a secondary dormancy, which in turn will lead to no germination. And this is important because malting barley has to be malted or germinated in order to be turned into beer. And as you can imagine, if you have a secondary dormancy, you won't have any germination, which stops the entire process in its tracks and makes people sad because you cannot make beer. So just a little bit more insight on what it is, how it's measured actually kind of helps conceptualize that. And so to measure it, you take 4 milliliters of water and 8 milliliters of water, put them in 2 petri dishes, and then add 100 barley kernels to each. The number of kernels that germinate in the 4 milliliter dish is taken to be the germination percentage. And then if you take the number of kernels germinated in the 4 milliliter dish and subtract what germinates in the 8 milliliter dish, you have the barley's water sensitivity. So in this little toy example, if you have 100 kernels that germinate within the 4 milliliter dish, and then 60 that germinate within the 8 milliliter dish, you have a water sensitivity of 40 for that malting barley line, or 40%. It's reported both ways. So moving right along to what's known about the trait. So malting barley water sensitivity is not something that has been studied a lot. It's kind of an open field. Some of the most recent papers upon it were actually published in the 60s. And the most recent review on it is from 1975, actually. And in that review, Brooks et al. States that storage conditions affect water sensitivity along with the genotype, the environment during grain fill, purportedly cool and wet weather increases water sensitivity, and then the time post-physiological maturity. So on top of this, there are some seed physiologists that have looked at this trait within barley in the early 2000s. And their eventual hypothesis was that hypoxia causes the kernel embryo to increase its ABA sensitivity, which in turn induces the secondary dormancy. And those studies were not done under the name of water sensitivity at all. They actually were published only in seed physiology books, and to the best of my knowledge, that hasn't crossed over into breeding at all. So moving right along, we set out to test 3 of these items, and those were the genotype, the environment during grain fill, and the time post-physiological maturity. To do that, we selected one of the populations that was going to be grown already. This was a regional malting barley trial. We selected a subset of 20 lines. There was 5 checks and 15 potential varieties or unreleased varieties. There was 2 locations, and each location had 2 replicates for each variety. And enough grain was sampled to take about one time point per month after physiological maturity for water sensitivity. And then there's some other questions that we're asking. Thanks, James. There's some other questions that we're asking with other populations, but these are not being discussed currently because we don't really have many results for them at all yet. So a little bit of data analysis. So we analyzed the regional plots with the following model: the phenotype being a function of the entry, the entry by location, and location and error, with location being the only fixed effect and the entry being the genotype. And then heritability was calculated in a pretty standard manner for a balanced data set. So the first thing that we were looking at, is there a genetic effect? And heritability is at least reasonably high for the water sensitivity trait. Let me get the laser pointer up here. You'll see, so this is, go back. All right. Heritability is reasonably high for the water sensitivity trait. Sorry, things are going wrong here. So we have, the laser pointer's not on? Why is it not on? So we have reasonably high heritability throughout the traits, and G is actually the germination percentage. It's called germination energy within the malting barley field, and that's high at all time points. Water sensitivity increases steadily in heritability, and we think that's actually because the actual dormancy interferes with the measurement of this induced secondary dormancy. So if we look at these first 2 time points for the germination percentage, we see that there is a lot of residual dormancy 11 and 33 days past physiological maturity, and that corresponds well with the low heritability water sensitivity. But if we drop those first 2 time points, we can still see that water sensitivity is changing over time. So we've got lines that have a great increase in water sensitivity in between 61 and 89 days post physiological maturity, and we have ones that are remaining reasonably constant. So with that, we conclude that water sensitivity does change over time. And lastly, we can see that water sensitivity is strongly influenced by the environment. And we only have 2 time points as of now, but it does appear to be a consistent effect. So we have Snyder always having a smaller effect on it than Caldwell. Interestingly enough, the location of Snyder was actually harvested about a month before the location at Caldwell, meaning that Caldwell actually had hotter and drier conditions during grain fill, which goes against the trend that was mentioned by Brooks et al. In 1975, that cool and wet environments contribute more to water sensitivity, have a greater water sensitivity effect. So with more data, we should be able to validate these trends, and that will be very interesting to see. So moving on to conclusions, we can conclude that time does change water sensitivity in this population. The environment does play a substantial role, and it's actually, the relationship is contrary to what has previously been stated in literature. And then the genotype does play a large role within water sensitivity. The effect of storage cannot be determined as of now. And then lastly, the future plans to design some experiments to test for sample and test for water sensitivity within the barley populations next year, to test this effect of storage as much as we can, to do a kernel number optimization, because as you can imagine, counting hundreds and thousands of barley kernels takes a while. So if we can decrease the amount of kernels we have to test at each time point, that would increase the amount of lines that we can get through the system. The further assays for these coming time points, you notice I only had 4 or 5 time points there. We still have 5 more to go. And then the testing of a large diverse panel and the associated GWAS for the genetic architecture of water sensitivity. And so that's what I have for y'all. I'd love to take questions.
Thank you, Travis. Yes, we have plenty of time for questions.
You mentioned cool and wet conditions influence what you think water sensitivity between your 2 environments.
Mm-hmm.
Do you have other ideas of any other physiological things that could help you?
So the question was, is there any other influence on water sensitivity apart from the environment? There's the storage conditions, which we haven't tested. It could be that humidity, the moisture percentage of the kernels influences the water sensitivity at the time that the test is being done. It's sort of up in the air currently.
I think this is cool research, but I figure if nobody's done a review of it since 1975, that must mean that maltsters aren't terribly interested in it.
This is true.
Why?
So the reason that is because malting has been, for the past 50 years, highly centralized. With the advent of small-scale malt houses, this is a trait that small-scale malt houses find very difficult to deal with, because if you've got a large batch process, you can pretty easily incorporate methods that will deal with the water sensitivity as is needed on a yearly basis, I would assume. And so it hasn't been a trait that has been published upon, but smaller scale malt houses really have a hard time dealing with it. The idea for this came from one of, so Aaron McLeod is a brewing chemist at Hartwick College, and he said that his input has been that water sensitivity is something that's very difficult for small maltsters to deal with.
I understand. So, like Anheuser-Busch, they would do small scale tests.
Yes.
Every barley batch that comes in, determine exactly the water sensitivity.
Yes, and then adjust
And then
their schedule accordingly. Yeah, that would be, so the statement was that Anheuser-Busch or other large malt houses would sample each batch that came in, then adjust the malting schedule accordingly, and that would be what I would assume would be correct, which also opens up an interesting avenue if they have all that data and they're willing to share it.
Would there be molecular biology or genetic ways of testing the abscisic acid hypothesis?
Yes, that's.
Okay.
I haven't thought too much about it. That's the seed physiologists that studied this back in 2006 or so. They studied the effect of ABA on it in more of a physiology and molecular biology standpoint, and so that would be something that I can draw on if I need to do that sort of test.
Do we have any questions on Zoom? All right, no questions on Zoom. Let's thank Travis one more time.
This has been a production of Cornell University, on the web at cornell.edu.